Hello, everyone. My name is Dr. Richard Horowitz, and I am the co-host of the Dr. Talk Healing Lyme Summit 2.0. My great pleasure today to introduce to you Dr. Jyotsna Shah. Dr. Shah is head of IGeneX Laboratories, who we have been using for, I think it's over 30 years at this point. We've been using IGeneX. And I'm so happy to have you on, Jyotsna, today to discuss the IGeneX testing for Lyme, for tick-borne relapsing fever, for Babesia, for Bartonella, for all the tick-borne disorders. So thank you for joining us today.
Thank you for inviting me.
Yes, of course. So tell me, Justin, how did you get involved? I mean, I know you have a PhD from Harvard. You discovered the fish testing.
No, I don't have a PhD from Harvard. My PhD was from Nairobi University, and I came to do fellowship at Harvard after.
Oh, it was the fellowship at Harvard.
Yeah.
Got it. Okay. But then you came to the United States. Your initial interest was in malaria. Is that what I've understood before? And then you kind of moved over a bit.
Malaria, basically tropical medicine. So I had done my PhD on schistosomiasis. And when I came here, I was involved in filariasis and then into malaria, TB, all infectious disease, kind of got into that. And then the choice was, do I go into genetics or do I continue with infectious disease? And while I was doing my fellowship at Harvard, Lyme disease was being talked about. It was pretty new, and they had just found it in Nantucket in white mice.
Mm-hmm.
And so we would hear about these studies that were epidemiology department was doing in Nantucket Island, which was close to Boston, and that they were finding these Borrelia in this white-footed mouse, and that was causing disease. It was becoming quite popular, and they started having ticks and trying to infect mice with that, and try and start growing cultures and things like, because there was a big animal house that Harvard had in tropical medicine, where they would grow different things, different small animals in which they will harvest different kind of parasites, filarial worms and things like that. Right. So I'd heard a bit about it and it sounded interesting. And then when I started work, then I decided to go into industry and not into academia for a very simple reason. I did not want to spend my weekends writing grant applications. I was seeing everybody there 7 days a week. And I said, no, this is not what I want. I want a regular life, 5-day, 2 days off, because I had a daughter that I wanted to spend time with. So I went into industry and I was thrown into all kinds of infectious disease, being asked to look into pneumocystis, which I really didn't know at the time what it was. So I had to go research about it. And I developed a whole set of probes. And this is where I started fluorescence in situ hybridization, because there was no other way of doing analysis of what I was finding in terms of designing probes. So that was the beginning of fish fluorescence in situ hybridization, and that technique proved to be very, very useful for pneumocystis. And that is how I further went into Babesia and did the fish assay. And after that, while I was there, there was one of the scientists came from Billy Burdolphy's lab, as it happened, and we used to share the office. So we would talk about things, and his whole family was infected with Lyme in Long Island. And so this kind of got, I got interested in that. And then when the opportunity came, either I could have gone to Stanford or they worked on tick-borne disease. I decided to join IGeneX and start working on tick-borne disease. And I'm still here after all these years.
And I don't think people know, but you and I go back quite a bit, because when I first discovered Babesia in the Hudson Valley back in the 1990s, we sent the ticks to IGeneX. You had discovered them right in the ticks, as had Rhode Island with Tom Mather's lab. And it was ultimately the fish testing, right, that was confirmed by New York State actually early on, that it was a reliable test. And that was, of course, my first experience with a patient in a wheelchair for 5 years who couldn't walk with Babesia, out of the wheelchair with Mepron and Zithromax. So, I mean, you and I go back actually quite a while using fish testing and helping IGeneX, right? Using IGeneX testing to get to the bottom of what was causing the illness in our patients.
Well, the credit goes to you because you gave us the samples to work on Babesia. You thought they had Babesia, and then we started working on the fish assay, and eventually we managed to get that assay approved. But not only that, that put us in what is Babesia? Started that project, and I think to this day, fish is one of the tests that everybody loves because it was designed at the genus level, so it detects species that we wouldn't find otherwise. And, two things that I would hear, one was from Dr. Fallon. He would say that every other test would fail, but fish would come back positive and a patient is treated just like you, patient gets better. So you guys had the experience, and I think with all your help, we were able to, push with the fish assay and get New York approval because you had talked to them in the
Yes.
beginning about Babesia. And, one time when we had a meeting at one of our meetings, I think the epidemiologist from New York State came to me and said, your results are wrong. There is nothing like this because some other lab had found that none of the ticks were positive for what we are saying. But the following year, he came back and I said, I apologize. That was really nice of him, but you were correct. What you found is what I am finding as well. So that kind of helped us to, say what we are doing has some merit.
Right. And this is the way science advances, right? I mean, we question things, we do experiments, and, you've been a part of the front lines. In my practice, I mean, I would never have been able to help the 13,000 plus people I have helped if it wasn't for IGeneX Lab. So I want to thank you and, of course, Nick, who passed. I mean, you guys have really been the front runners. And what's exciting for us, you just got FDA approval, which is groundbreaking, right, for getting the immunoblot approved for even home testing. Tell people about that a bit.
Okay, so what we have done is we always believed, as you know, that the standard two-tier testing wasn't the best way to go about testing for Lyme disease because the markers especially were missing that we knew were critical, and we always had shown that they were important by PCR, by, based on the clinical histories that you were treating patients with when they would get better when we reported out those results. So we said, if we are going to get the FDA approval, we really need to get the right test approved. And it doesn't matter what it takes. So what we decided to do was CDC was also very helpful because they have these panels that they provide whoever is doing research. So you start out with one simple panel, then you go into the next one and the final one. So you really look at about, I would say, 400 or more than 400 samples
Mhm.
to develop your test. So we started doing that when we had full confidence in the immunoblots and, had done enough by then. And we started testing and the results started appearing really good. We were, our sensitivity and specificity were outstanding. So then we said, well, I think we have got what really makes sense. So let's apply to FDA for the test. So we did apply and we cleared the IgG test. It is the most sensitive test on the market. It detects all the species that we currently know of in US and also in Europe and Australia. And it's a test that if we find something new, we can add on. And of course, we would have to get approval from FDA for the additional markers, but that's something that's possible. So, and the other question was there was this two-tier idea up to now that you had to do a two-tier test to get specificity. That was the main reason, but by doing that, you are losing sensitivity. And we
Mhm.
said, no, why can't we do a test that is a single test, not a two-tier test, a patient can do and get the right answer. So we have been working on it, and that is what was clear for IgG. And IgM, this is really interesting. And I think as a physician, you can, enlighten us on this. Our understanding is that IgM converts to IgG. That's the standard procedure. So when we are looking at patient samples, when we first got the results and we compared them with what was expected, we said the results make a lot of sense because early disease were most of them were IgM. Then you will see G and M together, and then you will see more Gs. And that is exactly what we are saying. The late stage Lyme arthritis, how would you classify it? Stage 3? So most of them in our hands were turning out to be IgG, not IgM. Whereas other tests that we use as a comparator really was not picking up IgG as well, but they were getting more IgMs. And to us, that didn't make sense. We said, well, I think our results are perfect. So we just went ahead and submitted that, and they came back, sorry, your sensitivity and third stage is low. We are not going to accept that. Can you improve? I said, we cannot improve with the test. This is the way it is. But if you look at G and M together, they're all positive in G because they've converted. And that is where they did not accept our argument. They said, we have to go by what is out there, and it's a comparator, so you don't match.
Yes. Well,
We are
in
not
my world, though, the IgM, and this, there was research done years ago at the University of California, Davis, showing that in the mouse model, right, when Borrelia invades the lymph nodes, it would get rid of the part of the lymph nodes that makes basically IgG and leaves IgM. And what we were seeing in our patients, and we replicated this and basically published it a few years ago, we got up to a 45% rate of CDC positive IgM blots in our chronic Lyme patients, not IgG. And our assumption at this point was because the immune system was being affected, just as it was in the mouse model. And that's why we weren't seeing as much IgG. But ultimately, because the immunoblot is recombinant DNA, right? The
right.
beauty is we don't have to worry about a false 31 band on a Western blot because of autoimmunity or Epstein-Barr, right? When we see any of the Borrelia specific bands on an immunoblot, 23 OSP-C, 31 OSP-A, 34 OSP-B, 39 very specific, 8393, I know in the right clinical setting, whether it's IgM or IgG, as long as it's fitting the clinical picture and we've ruled out other diseases, we are dealing with chronic Lyme. But
Right.
it is true, I do see much more IgM than IgG in our chronic Lyme patients because of the way the immune system's been affected.
That is true. We do see that, and that that happens even today. That's not what I'm saying. We do see IgM, but what I'm trying to make a point is that I believe when we were able to show very clearly that IgM is being picked up by the IgM blot and IgG is being picked up by the IgG blot, where we saw all the samples for only Lyme arthritis I'm talking about. I'm not talking about any other form, that I believe the results were correct, that they were really IgG and not IgM as well, or mostly IgG, very little IgM. The difference was when these tests were done by other groups, that they use tier 2. And as tier 1, what they use is total IgG and IgM, total antibodies. So that tier 1 is G and M, and there are some antigens to which antibodies are present for a much longer time in M form. And that is why they were getting positives. And when we were being very strictly IgM, we were missing some of them. That was the explanation. But the results that we presented as IgM specific were correct, but were not acceptable to them. So that's something we are working on, because we really believe that test is very useful. IgG, I think, is the best one, because we are able to include 31 and 34 in them, and all 93, as you say, 39, 23, 31. And we also have the CDC bands in there, but we don't read them. We provide the result both ways, but in our hands, it showed that that wasn't even necessary to call it positive. The hygienics criteria was working very well, and that is what's approved as well.
Right. So the point is people now have access. They have access to your test at home, right? For diagnosis.
Yes.
And I think that's really one of the breakthroughs. And, when we do an IGeneX panel, when someone comes to me for the first time, my standard is usually I'm going to get an IgM, IgG immunoblot for Lyme. I'm getting an
Mhm.
IgM IgG immunoblot for Babesia. I'm getting an IgM IgG immunoblot for Bartonella. I'm going to get a Babesia fish and a Bart fish. If I usually do that, and occasionally tick-borne relapsing fever and the rest, I find that I'm able to pick up really the vast majority of these infections in people. And although sometimes the fish can be negative initially because the bugs are hiding, ultimately, I would say the fish testing has been amazing in picking up Babesia because some of our patients are immunosuppressed. They don't make antibodies. The fish is the only way to pick it up. And I had invited you and Nick years ago to China. We got a call from the
Yes.
Chinese government. I mean, this was an amazing experience I'll tell people about. It was like 15 years ago. I was in France lecturing to infectious disease doctors in French, of all things. And I got a call from the Chinese government. They thought they were discovering Babesia, in epidemic fortune. And they asked me to come over. I wasn't sure if it was real. It turned out it was. They paid for our flights. And I invited you and Nick to come along with fish testing. We all met in
Yes.
China and proved, in fact, that it was Babesia that they were seeing. And I mean, it was really what I would say one of the great experiences of our lifetime. We were all right kind of together with the CDC,
Right.
National Academy of Sciences. I mean, it was really a great exchange of scientific information.
I think so. And, when they found out it's Babesia, since then there are a lot of publications that have come out. And one of the interesting things that nobody really knows because we haven't talked about it is that we are able to detect with the immunoblots species that they are discovering there because we have had cases which were Lyme genus positive, but are not Microtia, Duncani, or Oducolia.
Mhm.
But they are different species, and now by sequencing, because we do culture, and if we're lucky, we get cultures to work. There's never a guarantee that the culture is always going to be positive, but when we do get it, we've been able to find that what is picked up on the immunoblotted genus is a different species of Babesia that we have just discovered. We don't know too much about it, but it's a different species and the immunoblot is detecting it. So the point is the immunoblot at the genus level is really good at picking some unknown species of Babesia that are causing disease in humans, not just Microtia and Duncani, just like the fish assay.
So is this possibly like MO1, KO1? I mean, is it some of those species? Or are we talking maybe some of the European species that we're not used to seeing here? Do you know? We don't know yet what they are.
Yes, it based on little information, we are trying to work this out. It looks like a Chinese, what was discovered originally in China.
Uh-huh.
It looks similar to that at this point. And there's some other interesting findings that we need to confirm. But, this was, to us, this was exciting that we were able to find by sequencing species that are being detected on the immunoblot.
Hmm. So this
And
means,
yeah.
right. So clinically, this means for someone. So, if, for example, I have a patient that comes in with malaria-like symptoms. They're coming
Uh-huh.
in with day sweats, night sweats, chills, flushing, air hunger, can't catch my breath, cough. I've ruled out hyperthyroidism. I've ruled out tuberculosis, non-Hodgkin's lymphoma, menopause, right? Hyperthyroidism, all the things, autoimmune disorders, things that would cause drenching sweats. And you
Right.
can't find it with a Babesia microti titer or Duncani titer. You're basically saying that immunoblot will pick up some of these other species that could be accounting for malarial symptoms, but where we're not picking it up on the standard testing.
Yes, that is correct because these are the samples we were worried. How are we going to confirm that this really a babesia? That is one of our questions because Duncan in Microti, IFA is going to be negative. They're going to be negative. So this is why we are pursuing methods to confirm these other species or at least say these are at the genus level because our goal is to get an FDA approval for the immunoblots, which I think is very important. In fact, we are trying to work with them and see how we can make this happen. It may take some time, but that's something we are working with FDA on. Have you had some input from them? as to what. And I think it's going to be a joint effort to get something like this on the market because there are no comparators. There's nothing like this that we can compare with. And it's almost like what we had done for malaria fish, which was completely new, and it was working with them as a team to get those approved. And we are hoping we can do the same thing for Babesia with them. They've been very helpful and are willing to listen to what we have to say. And they have some really good scientists there that can give some advice and their thoughts as well. So
And
we'll
that's a
see how it
wonderful,
goes.
and Justin, what's wonderful to hear about this is that I think a lot of times people have thought like, oh, it's not easy to work with the CDC or the FDA, the rest. Now, when I worked on the first round of the tick-borne working group, my experience is these were wonderful people. We may have had our differences in
Right.
some things with, I don't believe this or that, but it was an open-minded scientific conversation going back and forth. We both listened. You're saying the same thing. I mean, this has been very helpful. It's not like adversarial. It was actually, let's work together, right, for the benefit of others. And
Right.
that's been your experience working with the FDA. And I actually had a great experience myself working with the governmental agencies when I was doing it also.
Right. Yeah, I think it's like nobody is going to agree to everything. We have to accept that science, that is science. We have our ideas, we believe it based on what we see, they believe what they see. And then we have to provide data, and every day we are finding new things. We might have said something 10 years ago, which now we would say is wrong. But
Mhm.
that is what we knew at the time. For example, band 31. We all thought on the Western blot it was important, and we were saying that till we found out that there was another marker underneath it that 50% of the samples were false positives. And that's why we developed a confirmation test for band 31.
Yes.
And with the immunoblot, that problem is gone now.
Yeah. And again, for people listening, and the problem with diagnosing the three Bs of Borrelia, Babesia, and Bartonella is that there are so many species out there. The problem is if you go to a local lab and you just get a Western blot, you're just basically checking one strain Borrelia sensu stricto. You're not getting, all of the sensu latu strains, including, as you were talking about, Abzelai and Guarini and some of the European strains, apart from California strains and the rest. And that's ultimately it's borreliosis, right? That's making people sick, just like
Yes.
Bartonellosis. You can't just do Bartonella henselae, Bartonella quintana, Bartonella bacilliformis. You need Elizabethae, you need Vinsoni subspecies, right? It's really the broad range of organisms that the immunoblots are picking up that make it so useful.
Yes. And I mean, going to Bartonella, we are seeing a species that it's not the standard for species that, really speaking, everybody is looking at Quintana and Hensley only by IFA. There are no other tests out there. And there's a lot of cross reactivity on the IFA. We have seen that. But on the immunoblots, we are picking multiple species that we would miss otherwise. Because we have tried to put 13 different species of Bartonella on the immunoblot.
Mm-hmm.
And we are seeing patterns. Sometimes, there's a cross between some species. Sometimes it's a very clear-cut answer you will get, but they are clearly Bartonella. The specificity is so high of this recombinant immunoblots. That's what we are trying to get through New York first. We are almost ready to submit our package. We've done everything that we need to do. Hopefully within less than a month, that package should be out because I just wanted all the confirmations around. And the team here, the research team is fantastic, and the lab people, I mean, they bend backwards to get whatever needs to be done in a timely fashion. So we think we have everything. And we are so lucky that we are able to culture everything in-house. The 4 species of Bartonella, we don't culture the other ones because we don't think it's necessary. And all the Borrelias, we have cultures, so we can confirm with the cultures as well, with the antibodies that we raise against them using these cultures when we want to do testing. So I think we've been lucky with everybody's help all over the world that we got all these collections here.
Right. And ultimately, even though New York State Department of Health early on, right, might have been difficult, ultimately they came around and said the test actually is accurate. We've gotten approval. And I think we're working more together now than I've ever
Yes.
sensed before. Now, a little bit what I'm seeing when people come in with these immunoblots, especially the tick-borne relapsing fever, talk
Yes.
also a little bit about this because it's a cousin of Lyme.
Yes.
It causes Lyme-like symptoms. How many of these species are you picking up on the tick-borne relapsing fever immunoblots?
8.
Eight of them also.
And yeah, so what we have found recently that Borrelia turriticata is on the rise.
Hmm.
And we've got that on the East Coast as well. We are very certain that that's what we are picking up on the East Coast. Another one we find is Turcica is quite common. People don't talk about it. They thought it's an animal species. No, it is human species. I mean, that was the first species we found on the Western blot was Turcica, reacted with Turcica, and in more Morellans patients as well, we had found Turcica like in Texas.
Hmm.
And we are finding Turcica miyamotoi is the other one. And we've had a few cases where we feel it's Parkeri as well. And, with the markers we are using, we can differentiate them. And sometimes we can't, but most of the times the patterns are becoming quite standard now that we understand what's going on with TBRF. And
Right.
we are finding that quite, maybe not that much on the East Coast as we are finding in Midwest and in California on the West
Mhm.
Coast. It seems to be higher percentage. The interesting thing about tick-borne relapsing fever is these patients don't have the classical symptoms of relapsing fever.
Yes.
And I think that is what is very misleading when we try to talk to people. They say, no, relapsing fever is relapsing fever. You don't need anything except PCR. What would you say on that?
Oh, I can tell you right now from my experience with it also. No, I mean, and you're right. I mean, what you're discussing is the classical textbook definitions.
Yes.
Like of babesia, you have to have hemolytic anemia,
Right.
renal failure, elevated liver functions, right? We don't see it.
Right.
I've only seen two cases, by the way, of hemolysis in my patients in all these thousands of people. And most of the time, it's in hospitalized patients who have congestive heart failure and other serious issues. That, by the way, that was the whole issue with the Chinese government. It wasn't looking like and smelling like babesia, but it's what you and I see, which is like, no, the textbook definitions, when you're mixing these organisms together, whatever you're reading in the textbook doesn't apply. It's true. I don't see the classic tick-borne relapsing
Right.
fever, relapsing sweats every couple of weeks. Right? We don't see that. What we're seeing in these subclinical presentations in people.
Yes.
So yeah, so it's you're right. It's a very different kind of presentation, especially when all of these bacteria and viruses and parasites are mixed together. It's
Mhm.
changing the way the body's immune system and even the clinical presentations of how they're presenting.
That's true. That's exactly what happened. So the initial cases we found here in California were we decided to just look because doctors would call me sometimes and say, "Hey, the patient has all the Lyme symptoms. Why is my patient not getting a positive test?" I said, "I can't make a test positive. A test is a test, and I can only tell you what the results are." So that got me thinking, what can that be? And then, of course, in the literature, they talk about relapsing fever being present. So we decided to look into that, and it started happening that these patients were really positive for relapsing fever and not for Borrelia. Very clear pictures, and we would look at multiple species of Borrelia to see if it's one of the other species that we don't have on the West, not are we detecting that, or are we detecting relapsing fever? And I've got lots that, and if I just show you, anybody who sees that has to believe that this is really true because we've got quite a few of them with all these 9 species of Borrelia on them and then multiple species of relapsing fever on the other one. And you compare the patient has one or doesn't have it. Some patients just have Lyme, some might have mixed infection. We've shown that by culture as well. But mostly all of them had similar symptoms.
Mhm. Interesting. And also with hard tick relapsing fever, Borrelia miyamotoi,
Yeah.
the people who are immunosuppressed usually are the ones who we know get right the worst symptoms with all of this.
Mhm.
But it's not easy. Although they say, you can get it on certain antibody tests or PCRs, we have a tough time picking it up right in the patients. And I've really only found that your tick-borne relapsing fever blot, right, is an easier way to pick it up. But These are soft ticks. So, I mean, do any of the patients actually say they've seen the soft ticks attached? Because, obviously in this part of the Northeast, we don't see it in the same way.
No, they haven't said it. Maybe it's for a short time for relapsing fever. The soft ticks are attaching, that we are not really seeing them, or if they are, they don't think about it. My other thought, I don't know whether this is true or not, because is it something in your locally, not, from which you're getting ticks? Like here, we have a lot of squirrels in California. Are they carrying the soft ticks that are causing relapsing fever? Who knows? Because
Yeah.
it's quite common. We don't know where they're getting from. Maybe there is a different host that we are not aware of.
Yeah, because I mean, when you look at the literature years ago, you would see like Borrelia hermsii infections in Arizona. Kids would go
Right.
camping in the woods in old cabins, and the soft ticks would be there, right? There were reasons why they were getting it, but
Yeah.
it's clear that these things are under the radar. Like a lot of these infections are starting to appear in people under the radar, and that's really the beauty of these immunoblots of being able to pick it up when you just don't suspect it otherwise.
Right. So I think it's important we are finding that. So we always recommend do both. If Lyme is negative, your symptoms up, you must go for relapsing fever. And that's something we are still, I mean, it's there, but like every other test, we continuously look at new markers to add on to that as we learn from the results, what we are seeing. But as I say, tachycardia was quite a surprise to start seeing from the East Coast. We, in fact, had a patient who called me from the East Coast, a good friend of mine, and she said she got a tick bite and she had a rash, classical rash. And this patient's the tachycardia cross reacts with one of the Lyme antigens.
Huh.
So she did the test and it came back positive, and I got her blood. She was positive for Babesia, not Microtia duncani, and she was positive for Taricates.
Hmm.
And what happened was it went to her eye. Her eye got infected. She called me and says, I got this infection and they've given me viral treatment. It's not working. I said, well, I've just looked at all your results. You got babesia and relapsing fever. You are not Microtia duncani. You have a different species. And sequencing confirmed that it was babesia species.
Yes.
And she had tachycardia on the East Coast in Massachusetts. This happened.
Right. So, I mean, we're learning over time, kind of. This is an
Yes.
evolving field, right? I mean, it's all
Yes.
all kind of changing over time. Are
Mhm.
there any other tests that you're doing right now for Migenics that you think you want to tell people about that would be
Yes.
important for them?
So the other test which we find quite useful is the culture test. And it's working really well. We got some data. This is like really, we thought it will only grow Borrelia. That's what we started out with. But once we started getting the cultures growing, we had a study going on for over 100 patients that we followed for months, up to 4 months. And we found that in some of them, so we said, okay, at 4 months, what are we going to do? The best thing is to pellet the cells and then make Western blots with that pellet. So whatever is there, we can put on a blot and then test it with different antibodies
Mhm.
that we had, rabbit antibodies to different pathogens. We did that for Lyme, TBRF, Babesia, and Bartonella. And when we tested that, we were able to detect antibodies, the antigens, just like on a Western blot. So
Uh-huh.
from cultures, we were making Western blots from patients' culture and testing them. And by that, you can see really that's something we're going to publish sooner or later. But there you can, if you have tested a patient's culture by say 10 antibodies, suddenly you might see one in some patient showing all the bands to one particular pathogen, like Babesia. We might see it to Lyme. We might see it to Holmesii, for example, to Bartonella. And we've got this data, which was very surprising because we thought we'll only be able to grow Borrelia and not Babesia. But Babesia grew, Bartonella grew. TBRF we expected to grow because Lyme and TBRF are similar. But that was very encouraging to see. And we are also seeing in real cultures, Rickettsia grows very well in our culture medium. But the most interesting thing is we are finding things that we would never think of when we culture. Like we found a different species of anaplasma in a patient that has only 2 cases reported from South America, platai. And the treatment is different. And when we send out for sequencing the one that was positive for anaplasma, it turned out to be platai. And we didn't know what that was. We searched, and it was from South America that there 2 cases. So we informed the doctor this was under research. And we found when we're doing Babesia, looking for Babesia primers cross-react with Clopopedia. It is, and this is something that there's a published paper where they were looking for Babesia in horses. So
Mhm.
using those primers, they did PCR, and when they sequenced, 50% of the horses were infected with Clopopedia.
And what about filaria? You see it over time in the literature, and I've always wondered whether some of these patients that, are negative by standard testing, it's not really, they don't really talk much about filaria, but it's in the literature. Are you finding like any of that getting in?
Uh, We haven't found any culture positive for Theileria at this point, but there could be reasons. Our cultures might not be working. They may not, but that's something we haven't found as yet in any of our patients. But Clopopedia, I mean, there were 2 cases reported from immunocompromised patients. We have already found 3 patients positive for Clopopedia by culture here in US. So that's something that is positive in patients that we are
And you
finding.
and you've gotten around some of the cult, for those of us who've been in the field as long as I have, over 40 years, you've gotten around some of these problems with the culture testing that was described years ago. You don't have the same problems that were discussed years ago with it.
No, we don't, because I think one of the things when you do culture, you got to be very careful how it is done. Contamination is your biggest problem. So the way we do it, this method is very simple. We are not using slides or anything where you would contaminate things, because the original culture method that was done by Advance, they had to put a special slide in it, coated with something so that it would work, but then you peel it off from there. So, the handling part was really messy for clinical use, the way it was done, and that didn't work well in our hands because we originally got the recipe from them, what they were doing under license, and we tried it for 2 years. In the end, we gave up and we said we can't do it because what was being shown, what they were calling positive, I had a hard time believing anything because on the IFA slides, every sample, I could not see a single thing that looked like Borrelia. And they were calling that positive. I said, if it is culture, you have to see a proper Borrelia in the culture. If you can't, then there's a problem.
Mhm.
And at the end, I said, I'm sorry, but I don't think your test is working. So we are giving back your patent,
Right.
all the expense you have had, that's fine, it's done with, but that's our answer. And we continue doing our own culturing because we were already culturing all the Borrelias, pure culture. So we said, why can't we see what will work? Well, we can play with that. When we did, and in fact, we run at least 3 or 4 tubes for each patient.
Mhm.
When we do culture, it's not as simple, it's pretty complicated. And
Got
then
it.
we run PCRs on it. That is why the test is expensive, because there's a lot of work that goes in to make sure that when we give a result, we know absolutely, we are sure that it's correct. We don't provide sequencing information because it's just too much, but we just tell them at the genus level and species level what we are finding.
So when would someone use, for example, you also have a lymphocyte transformation test, an ELISpot
Yes.
test, culture. When would someone decide? Because in general, I have found the immunoblots and fish testing to be most of what I need. When would somebody need to do a culture test or an ELISpot with a lymphocyte transformation test?
I think when they are immunocompromised, for example, they're going to be negative on the antibody test. That is when it's useful. Sometimes people want to know species or, then it's useful. It's not something I find the immunoblots give and immunoblots give you most of the information. Then if you want to know, some people are taking some medication and they want to see if it's cleared. That is when they're using culture to see if it's cleared out completely or not.
Okay.
So you can use it there, and some people just want a confirmation of what it is, so they'll order it. Now, what's very interesting is most people think that you just need to order tests for Borrelia. When we are culturing, a lot of times what we found is the patient is negative for Borrelia, but positive for rickettsia, for example, of in a plasma, and people don't think about it. Those are the things we are finding. I don't know, rickettsia seems to be quite a common problem. And we are finding different species of rickettsia in our cultures,
Uh-huh.
which is kind of interesting because we never were thinking of doing rickettsia, but it happens to grow extremely well in our culture medium.
Got it. So, and of course, we're not just talking Rocky Mountain spotted fever, rickettsia,
Um,
rickettsia, Norq fever,
yes,
cocktail of Burnetti. You're talking, or even typhus. You're talking other species of rickettsia, like you're talking even like the Swiss agent that they had discovered in Europe years ago.
we haven't found that, but we don't always go and sequence them because that was what we do for research. But now for clinical samples, we just report it as rickettsia.
Yes. Okay. That's very,
So we
very
don't, yeah.
good. So it's mainly these people that either you want to prove, like with by culture, that we've really eliminated it apart from clinical symptoms. Immunosuppressed patients, you could use the LTT, but otherwise, for the most part, what I'm doing with the immunoblots in fish, I'm finding is really going to be the majority of the
Yes.
testing that we need.
And the other test is the T cell test. Now,
Hmm.
looking at the data that we collected, we found that it's useful in patients who are literally negative by immunoblot. So again, immunocompromised or people, in the stage where very, very early stage, we find the ELISpot works extremely early or late in the disease it works,
Hmm.
where the patient has very little antibodies. But when the patient has lots of antibodies, T cell test does not work in our hands. That's what we saw, that patients who are positive by T cells had very low amounts of antibodies. Most of them were negative by immunoblots.
Got it.
Some might have been positive by PCR. That's what we found. Now, there's something very interesting that we published very recently. This is to show the importance of OSPE in
Mhm.
patients, all stages of Lyme disease. Usually if somebody gets a second infection, one of the first markers that shows up is band 31. It's antibodies to OSPE. And we did a study where we had looked at patients just on Western blots at the time and who were PCR positive. It was very interesting observation that a lot of them had 31 band in them. So we recently did a study with immunoblots, took those PCR positive patients and ran them on immunoblots. And we found that a lot of these patients were positive on the immunoblot for OSPE antigen from different species.
Mhm.
And they didn't have any other markers. This was very interesting. Only thing we saw was OSPE. A few had OSPE-C that we consider to be early disease, probably with IgM, but a lot of them had only OSPE. What we are talking about late stage, maybe chronic form patients who were having it very few bands, but they were positive either on IgG or IgM, one of them. But very interesting that we were able to show that importance of 34 in all these patients.
So is this going to be a problem like it was years ago with the VLA-15, the new Lyme vaccine that's coming out, which earlier on in the Dearborn, they took out the 31 and 34 bands because they didn't want them cross-reacting. Is this going to be another issue again we're going to be dealing with now of not being able to tell whether it's a vaccine-induced OSPE, or it's from an actual infection? How is this going to work?
That's going to be very interesting, right? Because 31 is OSPE is OSPE. And this is what we are seeing. So it's always bothered me when it comes to 31 and 34 being used as vaccines because patients also have antibodies to this even in late stage. I mean, that is why we wanted to show the importance of 31, and we know 34 is equally important. Um, just like 31. In fact, 34 might be even more important than 31. That's our preliminary data that we have collected as markers. We will be talking more about it once we know exactly what's going on that.
So we may, I mean, I wouldn't necessarily say this, and it's not in my population,
Yeah.
but in people who are thinking of getting the VLA-15, this new Lyme vaccine, they may actually want to get tested and find out what their immunoblot looks like before they actually take the vaccine, so they understand what their banding pattern is before and afterwards, because otherwise it
Yes.
might be a little bit confusing.
Yes, and again, it's going to depend on what test they're doing.
Hmm.
Because if they do the standard blots, 31 is never there, so that you never see 31 and 34 on any of those blots.
Right.
So that's going to be a big I think that is one of the problems that we've had forever.
Yeah. Yeah. No, this is fascinating. Shasta, this was great. Do you have anything else you'd like to add to just tell people? I mean, I think we've gone through a lot of the testing. Anything else you think is important to share with people?
No, I don't think so at this point. I think we've covered mostly everything.
Great,
And the
great.
only thing I have to say is thank you to all the physicians, all the patients who have been supporters. And I think it's the team. As a team, we have made progress. There's no way one group or one person can make this difference that we have made for the Lyme and other tick-borne disease patients. So I would like to thank everybody, and let's all continue working together to even, continue answering questions that come up.
Yes. No, that's great. And I agree. And honestly, IGeneX has been a godsend. From the very beginning of my clinical career, I could not have helped the thousands of patients I've helped without IGeneX. So I want to personally thank you from the bottom of my heart. I did tell this to Nick when he was around. You guys have been great. You've been the front runners of the lab testing, really helping with this worldwide epidemic. And it's been wonderful working with you and publishing with you and doing everything. And I really look forward to great things ahead. And in fact, when we finally get a Dapsone trial, a randomized trial, I look forward
Yeah.
to working with you on that trial also.
Oh, there's something I want to mention, and I
Yes.
need everybody's help on it
Yes.
because we have developed a home lateral flow assay for Lyme. Okay, and it seems to be working very well. The idea is to put it in doctor's office in the beginning with a reader, so there is no subjectivity in it. The
Hmm.
preliminary data is very encouraging. You do a finger prick. It requires 5 microliters of whole blood. You put it on the cassette, walk away with 3 drops of the buffer, walk away after half an hour, stick it in the reader and get an answer yes or no. And it
Oh,
seems to be quite a good marker, and that would help just to say, like a screening test.
yes.
And then, of course, all the other things would have to be done, but that's something we are developing. We thought that in some, eventually to be able to use at home as well. The data looks interesting. We are ready to start our clinical trials now, and we are looking for people to help us collect those patients and provide us the data.
That's great. And of course, because we expect, many thousands of people to be watching this Lyme Summit, for those of you who are listening, if you'd like to participate and help with this very, very important study, what is the best way for them to contact you, Jyotsna, to get in touch with you?
I think the best way is if they can send an email to IGeneX and say they want to participate, because it's only going to happen in doctor's set offices, because we can't do this with tens of hundreds of sites because of the IRBs and all the regulations that we have to follow. We will be helping the doctors get the IRBs, and we are looking for doctors who can give us at least 100, 30 to 40 positive patients at least, so that we get the numbers that we require to get to file for FDA.
Great.
But
So what exactly is the best email exactly to get in touch with you?
okay, jshah@igenix.com.
I'm sorry. So spell it out for people again.
J for Jones, SHAH at IGeneX, IGENEX.com.
That's great. Shasta, thank you so much for taking the time today. This was great. For those of you who've been listening, we've been interviewing Dr. Jasna Shah from iGeneX. She's been on the front lines with Lyme immunoblots and Lyme diagnostic testing for decades at this point. I want to thank you for taking your time. And again, for those of you who've been tuning in, my name is Dr. Richard Horowitz. I'm co-host for the Healing Lyme 2.0 Summit with Doctors Talks. Thank you for joining us. I look forward to seeing you again in another episode soon. Bye-bye.